So we are going to answer some biology questions which I have with me here. There are about 100 questions. Let's start. Question 1. What is the role of tRNA during translation? So what role does tRNA play during translation? So what we know is that tRNA, this is the molecule which is responsible for transferring amino acids, a target amino acid for that matter, from the cytoplasm to the ribosomes to create a polypeptide chain. That is the role of tRNA during translation. So A is saying it bond to open DNA. So just there, tRNA does not open the DNA strand. B is saying carry ribosomes. So tRNA does not carry ribosomes. C is saying it break apart mRNA. So tRNA doesn't have abilities to break the two strands. Then D is saying it carry amino acid to the messenger RNA for correct placement into the protein chain. So this is the correct statement with regards to transfer RNA. So it carries amino acids to the ribosomes. This is where the mRNA is at this point. So you have the small and the large subunit, then in between you have the messenger RNA. So the ribosomes will be able to read the base sequences on the mRNA. Then the tRNA responsible for a particular amino acid is going to go into the cytoplasm to go pick that amino acid. So that's its function. Then question 2 reads, how many codons are needed to specify 3 amino acids? So what is a codon? A codon is a triplet base. That is, if you have 3 bases, let's say AUG, this is a codon. So how many codons do we need to specify 3 amino acids? In short, we need 3 codons. That is going to be 9 bases. Let's say you have AUG, 1 codon, another one, let's say ACC, the second codon, then GUC, that is the third codon. So these are going to be 1, 2, 3. So we have 3 amino acids, 3 codons, 9 bases. So the correct answer here is we need 3 codons to specify for 3 amino acids. If the question was to be changed to say how many bases are needed to specify 3 amino acids, the answer is going to be 9. Because you have 9 bases, 3 amino acids, each with 3 bases. Question 3. One similarity between DNA and messenger RNA molecules is that they both contain. So what is the relationship between messenger RNA and DNA? So A is saying they have the same sugar. So this is false because mRNA and DNA do not have the same sugar. DNA has got deoxyribose, whereas RNA has got a ribose sugar. So it's not true. B is saying genetic codes based on the sequence of bases. They both have genetic codes based on sequences of bases. So we can pause for B. C is saying nitrogenous base known as uracil. That is, both they contain nitrogenous base known as uracil. So uracil, you can only find it in mRNA or in RNA. All the types of RNA can contain uracil, but not DNA. So it's false. D is saying double-stranded polymer. That is, both messenger RNA and DNA have got they are double-stranded polymers. This option will be somehow true, but it's not always. The reason being is because messenger RNA sometimes can be double-stranded molecule, but it's not always. Most of the time, it's a single-stranded molecule. So if we go with option D as the correct answer, it would mean that we are saying that mRNA is always a double-stranded molecule, just as DNA, which is not true. So this will be disqualified. So the correct answer here is going to be option B. That is, both messenger RNA and DNA have got genetic codes based on sequences of bases. So what we mean by this is that you know to say that you first need to have DNA. Then from DNA, that's where after transcription you have RNA. So in short, you cannot have messenger RNA without DNA. And the bases on the mRNA, the genetic code, is based on the bases that you are going to have from from DNA. So that is the relationship between the two. So the correct answer there is option B. We'll move on to question 4. So question 4 reads, which among the following is incorrect about the nature of genetic code? Codons are. So we're looking for the option which is false, meaning that 3 of these are correct. So which option is false? So we are saying that option A is saying that the genetic code or codons are universal. By universal, what do we mean? By universal, what we mean is that a particular amino acid will specify for the same amino acid, be it in plant cells or animal cells. Let's say we know that AUG specifies for an amino acid methionine. So it will specify for methionine in animals as well as in plant cells. That's what universality means. So it's true. Then B is saying overlapping. So I'll come for this to explain. Then C is saying the genetic code is commaless. So commaless, that is the genetic code doesn't have commas. If I give you a base sequence like this one here, there are no commas there. So it's true. The genetic code is commaless. There are no punctuations in short in the genetic code. Then D is saying the codons are triplet. The codons are triplet, which is true. A codon is a triplet base. AUG is a single codon. This one also, CCA, is also another single codon, which is true. The codon are triplet. So B is the false statement here. Why? It's because it's saying the codons are overlapping. The codons are non-overlapping. What does it mean? If we have a base sequence, these bases are read sequentially, they come in the sequence. So if we say it's overlapping, meaning it would mean that this A can be, you can have A here, then let's say C there. So making it A, C, C. That is what overlapping means. But they are not read like this. How they are read sequentially, they do not overlap. So you're going to read first 3, AUG1 codon, the second 3, another codon, and another set of 3, just like that. They are non-overlapping, they are not overlapping. So B, the false statement with regard to codons. 5 reads, in translation, dash is not an essential component. So what do we need for translation to take place, and what is not important? So we need amino acids. So that's true. During translation, you need amino acids. Okay, because you want to form a polypeptide chain. A polypeptide chain is a chain of amino acids, so you need them. Then B is saying ligase. So ligase is not needed in translation. This ligase, we know that this is an enzyme which is required to form the phosphodiester bond on the lagging strand, that is during replication. So you don't need ligase here. There's no formation of phosphodiester bonds during translation. So already there, option B is a false statement. So that's the answer. Messenger RNA, you need messenger RNA because that is what contains bases which are going to give you the polypeptide chain. Then anticodons. Anticodons, these are bases which are found on the anticodon arm of tRNA. Why are they important? They are important because they specify an amino acid which is going to be picked in from the cytoplasm. Let's say you want to pick an amino acid, methionine, AUG. This should contain anticodons which are complementary to these bases. So remember that this is within mRNA. So A will pair with E. Here we should have U. Then A, there we should have C. So this is an anticodon found on the anticodon arm of tRNA. It is very essential. It's the one which determines which tRNA is going to pick a particular amino acid. So option B is the false statement. We'll move on to question 6, which reads, this identifies a particular amino acid and its cognate tRNA molecule. This identifies a particular amino acid and its cognate tRNA molecule. So we have topoisomerase. Here we are still dealing with translation. So what identifies a particular amino acid and its tRNA, a cognate tRNA? This is a specific tRNA that can pick that particular amino acid. So what determines? So option A is saying topoisomerase. This is an enzyme which is found where? In DNA replication. So we don't need it during translation. This I think was supposed to be messenger RNA. So messenger RNA does not identify an amino acid. Just know that what identifies an amino acid or what picks an amino acid is transfer RNA. Ribosomes also cannot identify an amino acid and a particular tRNA to be picked. So we have transfer RNA synthetase. So this is an enzyme. So this is an enzyme which is responsible for identifying an amino acid to be picked from the cytoplasm, and also a specific tRNA which has got anticodons to that particular amino acid. So we need this enzyme, tRNA synthetase. It is an enzyme which is responsible for identifying an amino acid to be picked from the amino acid, and also the tRNA which is going to pick that particular amino acid. So that's the answer. tRNA synthetase. You need that enzyme. Question 7. Question 7 reads, protein synthesis corresponds to the process of. So we're still dealing with translation. Protein synthesis corresponds to the process of duplicating required DNA for synthesis of protein. So the process of duplicating required DNA for synthesis of proteins. So no. So during translation there is no duplicating of DNA. So it's a false statement. Then option B is saying formation of amino acids from messenger RNA. So protein synthesis corresponds to the process of formation of amino acids from messenger RNA. We'll pause here. I'll come back to explain. C is saying formation of messenger RNA from DNA template. So this is going to be transcription, not translation. Here we're looking at protein synthesis, which is translation. It's not transcription, so it's false. Then this information of amino acids from DNA template directly. So there's no direct formation of amino acids from DNA. DNA first has to be transcribed into RNA. Then RNA is going to now give us what? Amino acids. That is the basis which are going to be on the messenger RNA. So the answer here is going to be option B, which is saying formation of messenger RNA from, option B, formation of amino acids from messenger RNA. So the process of protein synthesis, it corresponds to the process or the formation of amino acids from mRNA. So you're going to have mRNA with its bases here. These bases on the mRNA are what we call codons. So these codons now are what we're going to form the amino acids. So the first three will specify a particular amino acid that is going to be picked by tRNA. Another set of three, just like that. Later on, these are going to be what? Joined to form a polypeptide chain. So that's what the process of protein synthesis corresponds to. Then we have question 8. So question 8 reads, if the mechanism of DNA replication in this bacterium were dispersive, what results would be found when the double-stranded, I think this question continues from here. Okay. If DNA was analyzed in the cesium chloride gradient after 2 generations in the medium which contain nitrogen 15, heavy nitrogen. So this question is coming from the Mendelssohn and the Starr experiment, which confirmed that DNA replication was semi-conservative. So in this case, we are saying if DNA replication was dispersive, what results were going to be obtained after growing this bacteria in a medium which contains heavy nitrogen, that is nitrogen 15, for 2 generations. So what are we going to see? So after 2 generations, what we are going to expect is that this bacteria at this point, it contains light nitrogen, that is nitrogen 14, nitrogen 14. Then you introduce it in a medium which contains heavy nitrogen. So heavy nitrogen is going to be nitrogen 15. So if DNA replication was dispersive, what would we expect is that we are going to have an intermediate or hybrid strands of DNA. That is, you would have the strands of DNA which contain both the old and the new strand, that is heavy and light nitrogen, something like this. So this was going to be a dispersive kind of DNA. If DNA replication was dispersive, we are expecting a hybrid strand of DNA. That is, each new strand should contain both old as well as a new strand in that in the same strand. The same with this other strand. It would have been dispersive. So let's see if we have an option which says a hybrid. So A is saying one band would be observed containing all the DNA. How true is that? So it's saying that one band would be observed containing all the DNA. So we pause there. Then B is saying two bands would be observed containing equal amount of DNA. How true is that? So what we are going to have is just within a single strand, you should have both old and new strand, that is light and heavy nitrogen in the same strand. So B is saying two bands would be observed containing equal amounts of DNA. By equal amounts of DNA, that is you are going to have two bands. So in cesium chloride, you would expect to have two bands containing equal amounts of DNA. Let's say down here you have heavy nitrogen, there you have what? Light nitrogen, something like that, which is not true. So after exposing this in cesium chloride, if DNA replication was dispersive, what we are going to expect is to have a hybrid, a uniform throughout. That is containing both heavy and light. C is saying two bands would be observed containing an equal amount of DNA. Then D is saying the DNA cannot form any band if replication is dispersive. So DNA cannot form any band if replication is dispersive. This is true. How is it true? It's because if we have a hybrid, what we would expect is something like uniform. Okay? A mixture which is uniform like this because it's a hybrid. So you'd expect something like this. No bands would be formed. This is dispersive. So the correct answer there is option D. Then question 9. So question 9 reads, during initiation of replication, A, DNA polymerase denatures adenine thymine-rich bases. So DNA polymerase does not denature, so it's false. What denatures is a helicase. Then B is saying replication begins when DNA protein binds the origin and the synthesized primers. So replication begins when DNA protein binds to the origin and synthesizes primers. So it does not synthesize primers. Yes, this molecule has to bind to the origin of replication, but it does not synthesize primers. So it's this what disqualifies it. What synthesizes primers is an enzyme known as primase. C is saying DNA B protein. So we don't have DNA B protein during replication, so it's false. Then D is saying two replication forks need to be created to establish a bidirectional replication. That is true. So during DNA replication, a bubble has to be created. Something like this. When you create a bubble, you have two replication forks. That is this Y-shaped here, another Y-shaped here. So this is a replication fork, that is another replication fork. So DNA replication is going to be bidirectional. That is, it will be taking place in both directions, this and that. So the correct answer there is option D. Then question 10. So question 10 reads, which function can be carried out by DNA replication proteins? So we know that during DNA replication we have got a number of proteins. So I think here it's about which enzyme has been correctly paired with its function. So A is saying topoisomerases wind the DNA into double helix. So topoisomerase does not wind, it cuts, or it eases the supercoiling which forms a head of replication fork. That is its function. Another name of DNA or topoisomerase is gyrase. So A is false. Then B is saying DNA ligase can initiate new DNA chains. So this is false as well. What can initiate new DNA chains is polymerase 3. C is saying single-stranded binding protein converts double-stranded DNA into single-stranded DNA. That is false as well. This can only be helicase. Then D is saying helicase breaks hydrogen bonds in the DNA. So this is true. The function of helicase is to break the hydrogen bonds, thereby separating the two strands. So that's D for question 10. 11. Which mechanism contributes to accuracy during DNA replication? So what mechanism contributes to accuracy during replication? This has to do with proofreading. So which enzymes are involved in proofreading? So we have both polymerase 1 and polymerase 3. These two enzymes have got proofreading activity with them. So that's what contributes to accuracy. Now let's see the options which we have. A is saying the mismatch repair system recognizes an incorrect base pair and corrects the mistake in the non-methylated strand. That is 11. So both strands are methylated. We don't have a non-methylated strand. So it's false. B is saying using primers increases accuracy. No. So the use of primers increases in accuracy. Why? It's because primers are made up of RNA bases. So in a case where these bases, let's say they have not been removed, it's going to result to what? To a base mismatch. Let's say uracil remains there. It's going to result to base mismatch. So it increases in accuracy and not accuracy. Then C is saying all DNA polymerases have a 5 to 3 exonuclease activity which can remove incorrect nucleotides during replication. So DNA polymerases have got the 3 to 5 exonuclease activity and not 3 to 5. Why are we saying so? It's because we know that polymerase can only add in the direction 5 to 3. So if it's to proofread, the proofread has to be done in reverse. That is, it should be in 3 to 5 direction. This is the exonuclease activity. That's what the exonuclease activity is all about. Then A is saying base stacking between nucleotides and the template DNA controls insertion of the correct nucleotide. Base stacking between nucleotides and the template DNA controls insertion of correct nucleotide. So this is false. Base stacking of nucleotides between the template DNA does not control insertion. So we have analyzed all the options. So B is false, C is false as well, D is also false. Option A was saying the mismatch repair system, that is DNA replication, has the mismatch repair system which recognizes an incorrect base pair and corrects the mistake in the non-methylated strand. Okay, so that's true. So the non-methylated strand, you know that during this process, during DNA replication, you are going to have an odd strand. This odd strand is the strand which is known as the methylated strand. Then you're also going to have a new strand. The new strand is a non-methylated strand. So let's say if DNA adds a wrong base, let's say we have uracil there, then it adds guanine instead of adenine. The DNA polymerase 3, which has proofreading, it will come back in reverse. then corrects this mistake, it will cut there and add the correct base, which is going to be adenine. That's how it corrects the mistake. So this is the non-methylated strand. The new strand is the non-methylated strand. So that option A is the correct option. We'll move on to 12. So 12 reads, a mutated E. coli cell contains a slightly altered RNA polymerase which does not function normally but still allows the cell to survive. So we have an E. coli which has got a mutated RNA polymerase but still allows the cell to survive. Which problem is unlikely to be the result of this altered RNA polymerase? Unlikely. So which problem is unlikely to be the result of this altered DNA? So it's A is saying some genes may be transcribed more frequently than normal. Some genes may be transcribed more frequently than normal. So we'll pause there. Let's check these other options. Okay, so this is a bit blurry. Then B is saying some mRNA molecules may be longer than normal. D is saying, so mRNA is based on the segment to be transcribed. So there will be nothing like it to be longer than normal. It will still be the same, the size will be maintained. Then C, the non-template strand may be transcribed in some genes. So the non-template strand cannot be transcribed. Then D is saying there may be an increased number of messenger RNA molecules with incorrect bases. Which is true. So here we're looking at the option which is unlikely to be as a result of the defect in RNA polymerase. So the altered RNA polymerase may lead to errors in transcription such as increased number of mRNA and the like. But some genes won't be transcribed more frequently than normal. So in short, all these three options are going to be as a result of what? Defect in RNA polymerase, but some genes will not be transcribed more than normal. So the correct answer here is option A. Some genes may be transcribed more frequently than normal. This cannot be as a result of a defect or a mutation in RNA polymerase. So A will not be as a result of that, whereas the rest of these will result from that defect. Then let me see, this page is blurry, so let's see if we can catch up. 13 says the codon UUU in the messenger RNA molecule results in phenylalanine being inserted as the protein is made. Which will be the characteristic of this codon? So what will be the characteristic of this codon UUU, which is phenylalanine? So we are saying that the tRNA molecule that binds to this codon must have the AAA anticodon. So we have this as a codon. So the tRNA to bind this RNA should have one codon. The base is complementary to that. That is this, that, and this. Okay? But it's not always because of Ubo hypothesis. So this can change. So let's check these other options. Then B is saying UU could code for both phenylalanine and alanine during translation. So what you should know is that a particular codon can only code for one specific amino acid, nothing like two. But a particular amino acid can be coded by more than one amino acid. So B is false. C is saying amino acid RNA synthetase for phenylalanine binds only this and that. So we have already said that due to Hubbard hypothesis, the amino acid, the tRNA carrying this amino acid may even be different from, may even contain a different anticodon at the third position there. So it's not always that the tRNA synthetase, in this case for phenylalanine, binds only to that. It's not always because of Hubbard hypothesis. Then this saying UU is probably only one of several codons that codes for phenylalanine. That is true. So phenylalanine is one of those bases that can be coded by more than one codon. So we have a genetic code here. Let's check how many codons specifies for phenylalanine. So here is the genetic code. So where is phenylalanine? So we have phenylalanine. So we have UUU, which is here. Yet we also have UUC. UUC also codes for phenylalanine. So it's true that UUU is just one of the codons which codes for phenylalanine. So the correct answer there is going to be option D. That is for question 13. Question 14, which reads, what is the size of the prokaryotic ribosome? So before we come there, what we know is that each ribosome, be it in eukaryotic or prokaryotic cell, has got both the small subunit as well as the large subunit. So I'll start with eukaryotic cell. The size of the small subunit in eukaryotic cell is 40S, where the large subunit in eukaryotic cell is 60S. But if they ask you for the total sedimentation size of the eukaryotic ribosome, you don't add to say 40 plus 60 to get 100, no. If they just ask you in general to say what is the size of the eukaryotic ribosome, the total is 80S. So you have to check out the question. Are they asking about the size of the small or the large subunit, or just in general? If they ask you in general, eukaryotic cell, the size is 80S. Then when it comes to prokaryotic cell, they also do have the small and large subunit, but the small subunit in prokaryotic cell is 30S. The large subunit in prokaryotic cell is 50S. The total or the same, you don't add 30 and 50, no. If they just ask in general, the size is 70S. So the total size of the prokaryotic ribosome is 70S. But if they are specific, asking about the small or the large, those are the sizes. So this question is asking us the general size of the prokaryotic ribosome. So the answer is going to be 70S. Question 15. Mark one which is not a stop codon. So we have 4 of these codons. So meaning that 3 of them are stop codon except one, and that is going to be the correct answer. So how many stop codons do we have? We only have 3 stop codons, okay? And what are these? So we have UAA, we have UGA, and also UAG. These are the only 3 stop codons which we have wherever you may go. You are angry, you go away, you are good. Something like that. That's how I remember this. So these are the only 3 stop codons. So a codon which is not among these, that is going to be the answer in this case. It's not a stop codon. So we have UAA is a stop codon, UAG is a stop codon, UGA is also a stop codon. Then this GGA is not a stop codon. So that's the answer in this case. Question 16. Question 16, which reads, eukaryotes differ from prokaryotes in mechanisms of DNA replication due to. So we're looking at the difference in which eukaryotic DNA replication differs from prokaryotic DNA replication. So A is saying eukaryotic DNA replication uses DNA primer rather than RNA primer. So this is not true. We don't have DNA primer, be it in prokaryotic or eukaryotic cell. What we only have are RNA primers. RNA primers, these are small base sequences of RNA. These are short sequences of RNA bases, and not we don't have DNA primer. B is saying different enzyme for synthesis of lagging and leading strand. That is, eukaryotic DNA replication has got different enzymes for synthesis of lagging and leading strand. I'll pause here. We'll come back. C is saying discontinuous rather than same discontinuous replication. So, what this means is that in eukaryotic replication, in eukaryotic DNA replication, DNA replication is discontinuous rather than semi-continuous. This is not true. Discontinuous, we have the lagging strand which is discontinuous. Then we have the leading strand which is what? Is continuous, the leading strand. So it is both. There is both discontinuous as well as the semi-discontinuous. Semi-discontinuous because you have the lagging strand which is discontinuous, then the leading strand is continuous. So semi simply means half. This is true both in prokaryotic and eukaryotic cells, so it's not a difference. Unidirectional rather than semi-discontinuous replication. So DNA replication both in prokaryotic and eukaryotic cell is bidirection. But that is, it takes place in two different directions at the same time. So it is not a difference. It will be taking place in this direction as well as in this direction, both in prokaryotic and eukaryotic cell. So the answer to this question is going to be option B, different enzyme for synthesis of lagging and leading strand. When you compare enzymes in prokaryotic and eukaryotic cell, one thing you discover is that eukaryotic DNA replication uses a complex of enzymes, which is not as compared to the enzymes in the prokaryotic DNA replication. So you have a number of enzymes that you need for replication to go ahead in eukaryotic cells. Okay, so it's a complex of enzymes in eukaryotic cell rather than in a prokaryotic cell. 20. So we had 16. I think the next question was supposed to be 17, but we have 20 here. So I can skip. Let's answer it. Which of the following is true about DNA polymerase? So DNA polymerase, we know that we have polymerase 1 and polymerase 3. So the most talked about polymerase is going to be polymerase 3. The function of polymerase 1 is that it removes primers and replaces them with DNA material. It also has got proofreading activity. Whereas polymerase 3, this is an enzyme which adds nucleotides to the new growing strand. It also has got proofreading activity. So if we look at this question, it has to do with direction. So this has to be polymerase 3. So we're going to discuss about polymerase 3. So polymerase 3, we are saying that this is an enzyme which adds nucleotide to the new growing strand. But one thing about it is that you can only add nucleotides in the direction 3 to 5 or 5 to 3 and not 3 to 5. So it cannot work, it doesn't work in this direction. It only works in one direction, that is 5 to 3. That is the reason why we have the lagging strand. The reason being is because this enzyme always tries to follow the 5 to 3 direction. Okay? So on the lagging strand, you find that the 5 will be here, the 3 will be there. And no. So we're going to have, on the lagging strand, you're going to have the 3 here, 5 will be here. So DNA polymerase 3 will be adding nucleotides in this direction. So it can only add nucleotide in the 5 to 3 direction. So A is saying it can synthesize DNA in the 5' to 3' direction. This sounds correct, but let's check if there's other options. It can only synthesize DNA, so the direction here, even here, okay? Then the last one, it can synthesize mRNA, so it doesn't synthesize mRNA, it synthesizes DNA, that is, it adds nucleotides of DNA to the new growing strand, that is synthesizing DNA in the 5 to 3 direction. Therefore, the correct answer for this question was option 1. Oh, that is option A. 17. 17 reads, what is the reaction in DNA replication catalyzed by DNA ligase? So what role does DNA ligase play in DNA replication? So DNA ligase is an enzyme which forms phosphodiester bond on the Okazaki fragments that are found in the lagging strand. So you know that on the lagging strand you have got these short DNA sequences which are joined. In between them you're going to have primers. So you have a number of primers. So you know that when DNA replication comes to an end, RNA or DNA polymerase 1 should remove these primers. Then you're going to have gaps which we call necks. So these gaps have to be filled. You know that this can be 5' end, this will be 3' end, 3' end, 5' end, 3' end, 5' end, 3' end, 5' end, like this. So at the 5' end, you know that you have a phosphate group. At the 3' end, you know that you have a hydroxyl group. So what this enzyme does, ligase, it joins these two, a phosphate at the 3' end to hydroxyl group at the 3' end to a phosphate at the 5' end, thereby sealing these gaps like that. So this bond which is formed there, that is what we call the phosphodiester bond. So option A is saying addition of new nucleotides to the leading strand. So when we talk of this enzyme, it is related to the lagging strand and not the leading strand. So this is false because of that. Then we check these other options. So we have okay, B is saying addition of new nucleotides to the lagging strand. So it doesn't add, it only joins. So this is wrong because of the addition. C is saying formation of phosphodiester bond between the 3 hydroxyl group of one Okazaki fragment and the 5' end phosphate group or the next on the lagging strand. So this is what I'm just from explaining. Then D is saying base pairing or the template and the newly formed DNA. So it doesn't pair bases between the template and the newly formed DNA. Its function is only to join the Okazaki fragments through a phosphodiester bond. So the correct answer is option C. 18 reads, during replication, Okazaki fragments elongate. So Okazaki fragments, as I mentioned earlier on, to say when you hear Okazaki fragments has to deal with the lagging strand. So here the statement which has the leading strand where they are going to be disqualified. So this already is disqualified because Okazaki fragments you never find them on the leading strand but lagging strand. So B has lagging strand, we can maintain. C leading strand. Then D also has got the lagging strand. So in short, A and C already disqualified. So remaining with B and D. So B is saying the Okazaki fragments elongate the lagging strand toward the replication fork. D is saying the Okazaki fragments elongate the lagging strand away from the replication fork. So here is the matter of does it elongate towards or away. So let me explain using this. You know, to say that you are going to have this replication bubble. So you're going to have the direction of replication. Here this is going to be the replication fork. Okay, so here you are going to have these bases which are linked through hydrogen bonds. Then you have helicase which is breaking these hydrogen bonds. So if this is let's say this is the 3' end, this will be 5' end. So the new strand on that where you need a primer here, then you're going to have a new strand which will be going like this. Okay, this will be the new strand in this direction. This is automatically, this will be 5' end, this will be 3' end. So remember we said that the new strands are synthesized by DNA polymerase 3, which can only add bases in the direction 3 to 5. So this is the 3 to 5 direction. Hence, this is going to be the leading strand. Now, what will happen down here? So down here, this strand, we know that it's going to be 5' end. This will be 3' end. So the new strand is going to have, you are going to have a number of primers on this strand. This will be primer. Let's say this is another primer. Then polymerase 3 will add bases in this direction. Why? It's because it's following the 5 to 3 direction. This one here, we're going to have, this will be 3' end, 5' end, 3' end, 5' end, just like that. Okay? So, what will be happening is that this helicase will continue breaking the hydrogen bond. Okay? So, as it is breaking the hydrogen bond, it is exposing more bases. So, let's say helicase has broken the hydrogen bonds. Now it has reached, let's say it's somewhere there. Another primer will be added. When this primer is added, let's say now it's added here, DNA polymerase 3 will come and join this, okay, and join that gap, just like that. Another primer will be added here, it will join, just like that. So the question now here is this lagging strand elongating away or towards the direction of replication, or towards the replication fork? So the answer is it's elongating towards the replication fork. Okay, it's elongating in this same direction. How? It's because the addition is taking place. Let's say you add here, it joins. You add another one there, it joins, just like that. So the elongation is going in this same direction, and it's not going away from the replication fork. So the correct answer there should be it elongates away from or towards the direction of replication, or towards the replication fork. So which option is that? So lagging strand towards the replication fork. So that is option B for this question, question 18. We move on to 19. 19, which reads, the fragments of DNA are joined together by which of the following enzymes? I think we're just from explaining on question 17. An enzyme which is responsible for joining the Okazaki fragments which are on the lagging strand is DNA ligase. So automatically that is option D, because it was already explained. 20. So we have another 20. Remember previously we had another 20. So I think that was just, but the question is different from this 20 we have here. So 20 reads, Tyler, Woods, and Hughes labeled Vicia DNA by allowing new DNA synthesis in the presence of radioactive thymine. After DNA replication, that is during S phase of the cell cycle, it was observed that so these were scientists, Tyler, Woods, and Hughes. These were scientists who did their experiment years after the Michelson and Starr experiment was conducted. We know that the Michelson and Starr experiment, which confirmed that DNA replication is semi-conservative, was conducted somewhere in 1958. So years later, these three scientists also were trying to confirm the findings of Michelson and Starr, if they were truly correct that DNA replication is semi-conservative. So with them, they labeled radioactive thymine, and after a number of duplications, what they discovered was that each chromosome, we know that each chromosome has got two chromatides. So what they discovered is that each chromosome had half labeled the radioactive thymine chromatide, the other half was unlabeled. So all the chromosomes had half labeled, that is half of their chromatide contained radioactive thymine, the other half didn't have these radioactive thymine. So they confirmed that the findings of Michelson and Starr, which confirmed that DNA replication was semi-conservative, was truly correct. It's because if in their experiment, if DNA replication was not same conservative, what they were going to find was that the entire chromosome was going to have radioactive thymine, or without radioactive thymine at all. But what they discovered is that half contained labeled radioactive thymine, the other half was unlabeled. So that is same, half labeled, half unlabeled, which is just as what Michelson and the Flynn discovered. So A is saying, what did they observe? So only one chromatide of chromosome was labeled. This is what I'm from explaining. Only half a chromatide or the chromosome, one of the chromatide of the chromosome was labeled, the other chromatide was unlabeled. B is saying both chromatides of the chromosome were labeled. No. C is saying both A and B. So for the fact that B is not the correct option, C can be the answer. Neither chromatide was labeled. So one was labeled, the other one was not. So the correct answer here is option A. So this experiment was conducted just to confirm the findings of Michelson and Starr that DNA replication was semi-conservative. 21. What is an operon? So before we look at the options, when we talk of an operon, this is a unit of genes which work together to achieve a common goal. So you need, let's say, if a particular molecule needs about, let's say, 4 enzymes for it to be done, then all these proteins, they are needed at once for that work to be achieved. So these cluster of genes or group of genes are what makes an operon. So an operon, you need a group of genes which work together to achieve a common goal. A group of genes can also be called a cluster of genes. That is an operon. So you can talk of like operon has got a number of genes here. It has got 3 genes. We have like Z, like Y, and like A. These genes have to be present together for the metabolism of lactose. If you talk of tryptophan operon, has got tryptophan gene A up to gene E. So it has got about 5. All these genes have to be present together for that operon to be successful. So that is what we call an operon. So A is saying a protein that binds to RNA segment and blocks the attachment of RNA or DNA polymerase, halting transcription. So this does not define an operon. B is saying a reversibly bound protein that enhances the transcription of a particular segment of RNA. So disqualified. This doesn't define an operon. C is saying a unit of DNA containing a cluster. A cluster is also known as a unit of genes under control of a promoter and a repressor. So this is what defines an operon. Then D is saying a protein around which DNA is coiled. So the coiling of DNA has to do with histones and not an operon. So D is also false. So the correct answer here is option C. Maybe one thing I didn't mention, there's a promoter here and a repressor. So a promoter, this is the region where RNA polymerase bind. So you're going to have these genes here. You know that for transcription to take place, you need, in this case, you need RNA polymerase. So RNA polymerase will bind to a region which is known as a promoter. So you need a promoter somewhere. Then you're also going to have a region which we call an operator. An operator, this is where a molecule known as a repressor binds. So you need both the repressor, which is going to bind to the operator, and the promoter where RNA polymerase bind. So option C, that is the correct answer for this question. 22. 22 reads, in a bacteria possessing the lac operon, which of the following occurs when glucose is low and lactose is abundant? So we're still looking at the lac operon. So in the lac operon, for you to understand lac operon better, you should just know the on and off theory. So the on and off theory is based on the concentration of these two molecules, lactose and glucose. Okay. So these are the two conditions which are going to guarantee whether the lycopene will be on or off. So we start with, in the presence of, if glucose is or lactose is present, glucose is absent. In this case, the lycopene is going to be expressed. That is, the lycopene is going to be said to be on. Why? It's because in the presence of glucose and absence of lactose, the lycopene is going to utilize, or this bacteria is going to utilize glucose. The reason being why bacteria utilizes glucose is because glucose, we know that is in its simplest form, is a monosaccharide. So there is no need for bacteria to break this glucose in its simplest form because it is already a simple sugar. Okay, so it's going to utilize this glucose, and there's going to be no expression of those genes. The structural genes are not going to be transcribed, hence the lycopene will be off in that case. Then in the case where glucose is absent and lactose is present, in this case the bacteria will not have an option. So it has to utilize this lactose. But lactose cannot be used in its disaccharide form. The bacteria has to find means and ways on how it can break down lactose into its simplest sugar, that is glucose and galactose, so that it can now utilize this glucose. So in this case, that's where now you need those enzymes which can break this lactose in these simplest sugars. Hence, the lycopene in that case is going to be on in the presence of lactose. The lycopene is going to be expressed because you need those three genes to work on this lactose to metabolize it. So this is where the theory of a lycopene is based on. So in this case, the question is saying you have abundance of lactose and low levels of glucose. What will happen in that case? So the lycopene is going to be expressed, or it's going to be on. So A is saying the beta-galactosidase acetylizes the operon. So in the presence of lactose, beta-galactosidase does not acetylize the lycopene. What beta-galactosidase does in the presence of lactose is that it breaks down or cleaves lactose into its simplest sugar, that is glucose and lactose. Then B is saying lactose permease cleaves lactose to glucose and galactose. So lactose permease, that is a protein which is produced by, it's a membrane-bound protein which is produced by a gene like Y. Its function is to allow lactose to get into from the medium to get into the cell of a bacteria. It's a membrane-bound protein, so it does not cleave. Cleave has to deal with beta-galactosidase because cleaving is cutting. So it is beta-galactosidase that cuts lactose into its simplest sugars, glucose and galactose. C is saying lactose metabolism is increased by lactose binding to the operon. This sounds quite close. Let's check D. D is here, which reads the transport of lactose into the cell is enhanced. That is, if lactose is present, the transport of this lactose into the cell is going to be enhanced. That is going to be the transportation of lactose. So we now have to check C and D, which one is more accurate. Lactose metabolism is increased by lactose binding to the operon. So C is not accurate enough because lactose doesn't just bind to the operon, it binds to the repressor. And by the way, it's not lactose direct which binds to the repressor. Lactose has to be modified before it binds to the repressor. It has to be modified to become allolactose. When it becomes allolactose, this allolactose is the one now which is going to act as an inducer that will bind to the repressor on the operator. Then the repressor will move out of the operator, thereby allowing transcription to take place. So D is going to be the correct answer here. The transport of lactose into the cell is enhanced. As we said, you are going to have lactose is present. So this lactose now has to be transported into the inside of the cell. That's why we have an enzyme, beta-galactosidase permease, that is membrane bound, which will allow this lactose to get where? Inside the cell. Excuse me. So D for 22. We move on to 23. excuse me. Okay, 23. The effect of allolactose is, so I bit tackled this. So allolactose, it induces conformation change of the repressor. What will happen first is that you have lactose. This lactose will get inside the cell of the bacteria. Then it has now to be changed into allolactose. This allolactose will go and bind with the repressor which is sitting on the operator. When the allolactose binds with the repressor will lose its binding affinity to the operator, thereby moving out of what? Out of the operator there, by allowing RNA polymerase now to transcribe the structural gene. So it induces conformation change to the repressor. That's the function of allolactose. So A is saying enhancement of binding of RNA polymerase to DNA, thereby increasing gene transcription. So it doesn't enhance the binding of RNA polymerase to DNA. This is what enhances the binding of RNA polymerase to DNA is the cyclic adenosine monophosphate under the cap. This complex is what enhances option 1. It's not an allolactose. B is saying repression of the lactoperon when glucose is present. So glucose has to be absent for this allolactose to be present or to bind to the repressor. When glucose is present, the repressor will be sitting on the operator and the allolactose won't be there, because the presence of glucose means that lactose is not present. Even if it's present, it will not be in use, so you not have allolactose. C is saying conformation change in the repressor protein inducing the lactoperon. This is accurate and true, because that's what we're from explaining. So allolactose will induce conformation change to the repressor. This repressor, when binds with the allolactose, it will move out of the operator, thereby allowing RNA polymerase to induce the lactoperon. So option B is true. D, which was saying dissociation of RNA polymerase from, so this at any way doesn't favor allolactose. So the correct option there is option C. 24. Which of the following is not true to the nature of genetic code? So we have 4 options, meaning that 3 of them are correct. Just 1 is not true. So A is saying codon is a triplet. So when you talk of a codon is made up of 3 bases. Let's say UAG. UAG, this is a codon, a single codon, is a triplet base. So A is true. B is saying codons are commaless. Commaless, that is, there is no punctuation in a codon. UAG, there are no commas to say U, A, C. There is no punctuation in the codon. So B as well is true. C is saying codons are overlapping. Doubtful, we'll come back. D is saying codons are universal. So when you say codons are universal, what we mean is that if AUG codes for methionine in plant cells, it will still code for methionine even in a animal cell or eukaryotic cell. So it's true, genetic code or codons are universal. So overlapping is a false statement here. The codons are non-overlapping. Overlapping, what it means is that if I give you this sequence, these bases are read sequentially, they come in the sequence here. They are read in pairs of 3, like that. So if we say the codons are overlapping, what it means is that you can have U here, then let's say you have another U there, then C there, forming UCA. So that is overlapping. The bases are like jumping, so they don't overlap. It's non-overlapping. So C is a false statement with regard to genetic code. So hence, in this case, is the correct answer. 25. Which of the following recognizes a specific amino acid and its cognate tRNA molecule? So this is a repeat of, I think, question 7. The answer is tRNA synthetase. You can go back to question 7. I explained there. It's a repeat. Then we move on to 26. So 26 reads, which of the following does not take part in gene expression? So before we look at the options, when we talk of gene expression has to do with transcription and translation. These two processes are what makes up gene expression, except replication. Replication is not part of gene expression. On these two are what makes up gene expression. So let's go back. What options do we have? A is saying replication. So I've already mentioned that replication is not part of gene expression, so it's false. Hence, in this case, is the correct answer. Which of the following does not take part? But let's disqualify these others. Transcription, yes, we say it is part of gene expression. RNA processing, that is protein synthesis, which is part of translation, so it's true. Translation, yeah. So B to C, they are part of gene expression. Replication is not, so the correct answer there is option A. 27. Which of the following is wrongly paired? So we have option A to D. I think this is with regard to bond formation. So proteins, we are saying that the bond which is found in proteins, the peptide bond, that is true. Nucleic acid is saying hydrogen bond, that is true. Talk of DNA, which is a double-stranded molecule. You have got two complementary strands. These complementary strands have got bases, and these bases are linked through what? Are linked through a hydrogen bond. So that's true. Even RNA also has got these complementary hydrogen bond, or complementary bases which are linked through hydrogen bond. Then we have C, which is saying polysaccharide. We have glycosidic bond. So polysaccharide, talk of how the sugars are linked to each other. So let's say if you have glucose and galactose linking together, you're going to have a bond formation where a condensation reaction is going to take place. When condensation reaction takes place, where there's going to be a release of water molecule, that bond which is going to be formed there is what we call a glycosidic bond. Then D is saying phospholipids is linked through a phosphate linkage. I think this is false. So what is a phospholipid? So a phospholipid, you have a glycerol molecule linked to a phosphate group. So this glycerol molecule has this OH group and you're going to have two fatty acids. So these fatty acids have to be linked to this glycerol molecule. So there's bond formation between this fatty acid and that hydroxyl group. This fatty acid which contains this group and that hydroxyl group. So this bond which is formed there is an ester linkage and not a phosphate linkage. So option D is a false statement. Hence in this case it is the one which is wrongly matched. Hence that's the answer. 28. In protein synthesis, translocation is initiated with a movement of. So protein synthesis, you know that you're going to have messenger RNA, the large ribosomal subunit, and the small subunit. The large ribosomal subunit has got three sites. You have the E site, the P site, and the A site. So translocation is a movement of one step. This ribosome is going to move one step, that is one codon forward. So when it moves one codon forward, what will happen now is that this codon will find itself in the A site. In the P site, in this case, we're going to have a tRNA. This tRNA has delivered an amino acid, so it becomes unchanged. Hence, it has now to leave through the exit site. So when translocation takes place, this A site is going to find itself on this next codon. Then what was in the A site will be in the P site. This transfer RNA, which has already delivered an amino acid, is now going to find itself in the E site. Hence, it will leave the ribosome through the exit site. That is a translocation. It will move, the ribosomes will move one codon forward, thereby allowing the unchanged tRNA, which has already delivered an amino acid, to find itself in the exit site and leave. So A is saying tRNA is a movement of tRNA from P site to A site. So the tRNA has to move from the A site, from the P site to the E site and not the A site. Then B is saying the movement of dipeptido tRNA from A site to P site. This is false as well. tRNA has to move from the P site to the E site, not A to P. Then C is saying the movement of tRNA from the A site to P site. Already explained there. It's not true. Then D is saying the movement of tRNA from P site to E site. So this is a true statement. tRNA has to find itself in the E site so that it can now leave. E stands for exit. P is peptido where the peptide chain formation is formed. Then A is acetylase or acetyl. 29. Protein synthesis corresponds to the process of A. Duplicating required DNA for synthesis of proteins. So false. There's no duplication of DNA when you talk of protein synthesis. B is saying formation of amino acids from mRNA. So this one sounds correct. We'll come back. C is saying formation of mRNA from DNA. This is transcription. It's not defining protein synthesis, which is translation. So it's false. D is saying formation of amino acid from DNA. Amino acids are formed from mRNA and not DNA. So it's also a false statement. Therefore, the correct answer here is going to be C. Formation of mRNA from, sorry, not C, but B. Formation of amino acids from mRNA. So you know that you have mRNA and bases here. So for you to form these amino acids, it will depend on the bases that you have here. So that codon, we have one amino acid. This codon also another amino acid, just like that. So you form these amino acids from the mRNA, which has sequences that will detect about the polypeptide chain to be formed. So option B for 29. 30. You have a piece of DNA which is a sequence with a sequence. So this piece of DNA has a sequence ATGC. Then what is the question now? So it's saying A is at the 5' end. That is, this A is at the 5' end. How to read that? B saying A is at the 3' end. How to read that? Then C is saying A could be either at the 5' end and at the 3' end of DNA. So one thing which we have to know is that DNA is a double-stranded molecule. That is, it has got two strands which run in the opposite direction. So if we have this is 3' end, that will be 5' end. Automatically this will be 3' end, this will be 5' end. So this sequence, if randomly I give you a sequence, like in this case we've been given A, T, G, C, we don't know whether this base sequence was extracted from this strand or it was extracted from this strand. So we can't really tell that this is going to be the 3' end. What if this DNA was extracted from this strand? It means that this will be 3' end or 5' end. So the correct answer here has to be option C, which is saying A could either be at the 5' end or at the 3' end, because DNA is a double-stranded molecule. So that has to be the answer with that regard.